Imagine a vast library filled with books containing the instructions to build and operate a human being. This library represents our DNA, and the books are our genes. But not all genes need to be active all the time. That's where DNA methylation comes in. DNA methylation is a process where a small chemical tag, called a methyl group, attaches to a gene. Think of it like placing a sticky note on a book's cover. This note doesn't change the words in the book, but it can signal that the book should be closed and put away. In cells, this sticky note can tell the cell's machinery not to read that particular gene. This process is essential for normal cell function, allowing different cells to specialize by turning on or off specific sets of genes. Without DNA methylation, our cells wouldn't know which genes to use, leading to chaos and dysfunction. It's a fundamental process that keeps our cells working in harmony. DNA methylation plays a crucial role in controlling gene expression, the process of using genetic information to create proteins and other molecules needed for cell function. By attaching to specific regions of DNA, methyl groups can effectively silence genes. This silencing occurs because the methyl group blocks the cellular machinery responsible for reading the gene's instructions. Imagine a librarian unable to open a book because of a sticky note. Similarly, the cell cannot access the information encoded in a methylated gene. This process is vital for normal development and cell specialization. For instance, DNA methylation ensures that a skin cell doesn't start producing liver enzymes, or a brain cell doesn't try to become a muscle cell. However, problems arise when DNA methylation patterns are disrupted. Like a library with misplaced books and inaccurate cataloging, the cell can lose its ability to regulate gene expression properly. This dysregulation can have serious consequences, including the development of cancer. Our bodies have a remarkable defense system against cancer, and at the heart of this system lies a group of genes called tumor suppressor genes. These genes act like vigilant police officers, patrolling our cells for signs of trouble. Tumor suppressors have various functions, including repairing damaged DNA, controlling cell growth and division, and even triggering programmed cell death or apoptosis when a cell becomes too damaged to repair. They are essential for preventing the uncontrolled growth and spread of cells that characterize cancer. However, these guardians are not invincible. Like any police force, they can be silenced or deactivated, leaving the cell vulnerable to dangerous elements. And one way this silencing can occur is through the disruption of DNA methylation patterns. Cancer often arises from errors in our DNA, mutations that can disrupt normal cell behavior and lead to uncontrolled growth. While some mutations directly activate cancer-promoting genes, others silence the very genes designed to protect us, the tumor suppressors. DNA methylation plays a critical role in this silencing process. In many cancers, researchers have observed abnormally high levels of methylation in the regions of DNA where tumor suppressor genes reside. This hypermethylation effectively shuts down these crucial genes, like turning off the security system in our cellular cities. With the guardians silenced, damaged cells can proliferate unchecked, leading to tumor formation and progression. For example, in some colorectal cancers, hypermethylation of a tumor suppressor gene called MLH1 is frequently observed. This silencing of MLH1 disrupts DNA repair mechanisms, making the cells more susceptible to accumulating mutations and transforming into cancerous cells. The discovery of this link between DNA methylation and cancer has opened up exciting new avenues for both diagnosis and treatment.